Thank you. Thank you so much for the introduction, and thank you so much for the invitation. It's really great to be here. It's a great meeting. It's a great city, so I'm very happy to be part of it. Thank you. Let me just put a laser pointer here. Laser pointer. There we go. Good. So, what I will be talking about today is single-cell epigenomics. So this is what my group is interested in. We work a lot with single-cell data. And I'm sure that this is happening to all of you very often, where we have collaborators that approach us and they say, we did a single-cell experiment. And they don't even mention it, but they obviously mean they've done a single-cell transcriptomics experiment. And our argument is always to say, hey, you know, if you look at the single-cell transcriptome, you're actually missing the regulatory component that is sitting there in every single cell between the genome and the transcriptome, and that allows to generate so many different transcriptomes from one and the same genome. I'm sure in this session, I don't need to convince anyone about that. And the other thing is that obviously by construction, single-cell RNA-seq is a gene-based approach, while with the single-cell epigenomic measurements, we measure everywhere in the genome, so you potentially get so much more information. So this is a very, very simplified plot here, where I try to just depict like the most usually used methods for single-cell measurement. There are, of course, many other measurements that can be used. So we can measure the genome of individual single cells with single-cell DNA-seq, and the most famous one, the transcriptome of individual single cells with single-cell RNA-seq. But then we have these layers in between, and I would say for chromatin architecture, we are mainly using single-cell ATAC-seq at the moment. And for methylome, there are different flavors of it, but there are different versions of the single-cell bisulfite sequencing that you can use to measure the single-cell DNA methylome. So what do we do in my group? We take these layers of information that are sitting there between the genome and the transcriptome, and we try to build methods that will allow us to understand how these layers work and what's then the interplay between these layers of regulation, the genome and the transcriptome of the individual single cells. And so for today, I will present you different things that you can do with this type of single-cell epigenomics data. I will tell you how you can use it in the most obvious way to study cell identity, how you can also use it to study DNA heterogeneity, but not with single-cell, but with single-molecule data, and how you can also use this single-cell epigenomic data to call copy number variations in individual single cells. So how do we use that data to study a single-cell identity? I'm sure you're very familiar with that. There are so many different methods out there that do that at the moment. I will just use it as a small introduction into the topic. So in my group, we have developed one method in particular, which is called episcampi for epigenomic single-cell analysis in Python. And for the ones of you familiar with transcriptomic data, I've used Scampi. So this is like the epigenetic version of it. It's embedded into the same pipeline. And episcampi can take single-cell DNA methylation data, can take single-cell ATAC data, and we build it already in a very modular way such that then you can go into multi-omics, for example, with ChIP-seq data with different histomodifications. And the work I will present was done by a PhD student in my lab, Anna Danese. So what's the first step that you need to do when you analyze single-cell epigenomic data? So the first, most important step is the feature selection. And when you analyze single-cell RNA-seq data, you do feature selection, but you do not even think about it. You take your genes and you take your cells, and you build a count matrix. What you've done is feature selection. You've decided, I'm looking at the genes. When you have single-cell epigenomic data, you have information everywhere in the genome. And the first question is, okay, how do I summarize this data? In our approach, we decided to be as generalist as possible, which means that we can either look at windows, how much methylation or how much openness is in every window in the genome, how much methylation or how much openness is in different annotations in the genome, for example, transcription factor binding sites, enhancers, whatever you may be interested in. Or, of course, if you have single-cell ATAC-seq data, we often use peak data, called in the bulk. Once you've decided your feature spaces, we quantify for you how much of every cell is open or closed or methylated and unmethylated in every feature, and then you get a bunch of count matrices. And then what you will, of course, do with these count matrices, you need to do some cleaning of them, some typical QC. But then basically what you have is a matrix that has the features and the cells. And with that matrix, you can construct, like with single-cell RNA-seq, a neighborhood graph. You have your KNN graph, for example, based on whatever distance that you're interested between the cells. And then you can do all the usual downstream analysis that require a graph. So you can, for example, do visualization, or you can do clustering, for example. And this toy data that I'm showing you here, it's not a toy, actually, it's real data that we downloaded from a publication. This is single-cell DNA methylation data from a bunch of neurons that was profiled already quite some time ago in 2017. So if we look at these clusters here, what I hope you appreciate compared to single-cell RNA-seq data, I'm going to blow it up, is that you can actually construct for the same single cells, you can embed your cells based on many different feature spaces. So here I'm showing to you the same single cells embedded based on the window DNA methylation or the gene body DNA methylation, the promoter or the enhancer DNA methylation. This is something that you obviously cannot do with single-cell RNA-seq data. Here you can really check how much the different parts of the genome are giving me information about the cell type, about what these cells are doing. And then you can compare. For example, you can say, how do the clusters between promoters and enhancers compare to each other? You can, for example, discover, oh, look, this cluster is split in two with the enhancers, for example, and not with the promoters. Or you can check which is the feature space that best describes your data. For example, here with the silhouette score, how much separate are the cell types among them? And so now this is kind of the classical still kind of annotation, gene bodies, promoters, enhancers. Now we are pushing that one step further by saying, you know what, actually a TAC-seq or DNA methylation give you information everywhere in the genome. So why do we even look at peaks? Why do we even look at promoters? We can look anywhere. And what we are doing now, I'm showing it to you here. This is an example with a 10x data set. So this is a TAC-seq. And we decided to partition it in many different feature spaces. Peaks, obvious. You have, this is PBMCs, you have all the different cell types in the different colors, you can identify them in the peaks. Other classical feature spaces, gene activity, how open is the gene and promoter, promoters, windows. But then we thought, let's try anything. I mean, why not tRNAs? What's wrong with the openness at tRNAs? Or what's wrong with the openness at repetitive elements? What's wrong with the, so what we actually discovered after doing some imputation of the data is that actually the majority of annotations in the genome contain a lot of information about the cell type that you are looking at. And now what we are working on is also like in more like unsupervised methods to actually be able to understand how much every one of these features is actually contributing to the cell type identity, and how much information we can get from the single-cell ATAC-seq data that you would really never be able to obtain from a single-cell RNA-seq data, because these features, you really do not recover them with the classical single-cell RNA-seq. Okay, so here we were here. We have all the different feature spaces. We have all the different cells. And then we can ask, we can do some differential tests. What cell is what? We can then obviously give them names with the collaboration with biologists. And of course, if we are not looking at separate cell types, but we are looking more like at the differentiation process, we can also do pseudotemporal ordering without problem with single-cell DNA methylation or single-cell ATAC-seq. And we have done in my group a lot of work on data integration, testing a lot of different methods that are out there for integrating data for batch correction. And we've tested them on single-cell RNA-seq and single-cell ATAC-seq, and this was already published a couple of years ago. It was a huge effort that we did together with a group of Fabian Thijs, where we did a benchmarking of many different Atlas integration methods for RNA and for ATAC. And the other pipeline was already published some time ago. Okay, so this is how do we do to understand cell type heterogeneity with single-cell data. And I told you that our pipeline works for ATAC and for bisulfite sequencing single-cell level. But I was actually kind of cheating a little bit, because what we get nowadays is single-cell ATAC-seq data. At the single-cell level, it looks like everybody forgot that there used to be a thing called DNA methylation that was very important. And now it doesn't matter. We have ATAC-seq. We don't need to do it anymore. But I think it's a pity that we forgot about the heterogeneity at the level of DNA methylation and how much this information is useful to us. So in the group, we decided to look at this long-read technology that we heard a lot about this morning, and how we can actually use that long-read information to extract clones at the DNA methylation level, which are present in the population of single cells. So imagine like this cartoon here. You have a sample, for example, a tumor, and this tumor has different cell types mixed inside of it. This would be like the blue, the red, and the purple cell types. And these cell types, they are defined by different genome-wide DNA methylation profiles. This would be this epiclone A, epiclone B, and epiclone C. And these lollipops are the cytosines. Yeah, a full lollipop is a methylated cytosine, an empty lollipop is an unmethylated cytosine. And it could even be that you do a chemotherapy, for example, on your cancer, and then it changes the heterogeneity of the sample, and you want to study that. Obviously, the best would be to do single-cell bisulfite sequencing. There you get your clones, you get your cells, you can get to like consensus profiles per clone and whatnot. But it's very expensive, and it's apparently extremely difficult to set up in the lab. So very few number of labs are doing that at the moment. What you could do instead, you can do a traditional Illumina sequencing where you have your 150 base pair long reads, and then per CpG in the genome, you can say, well, I have a 70-30%, I have a 50-50, whatnot. But it doesn't really tell you anything about the longitudinal information of these cells. What we thought is that we could use this nanopore long read data where the reads are around 1,000, more than 1,000 base pair long. And there we can use this spatial information that links the different cytosines which are sitting on the genome, and it links them on the read. And we know that one read is coming from one cell. So every read is a proxy for what the individual cells look like at the beginning. So then what we can do, we can take this long read data, we can cluster it somehow, and we can then extract what are the different methylation clones present in the data. So this is what we've done. This is a PhD student that did her PhD in my group. She's now at the Charité in Berlin. And what she did is that she developed a method that we called epidelo for epigenetic deconvolution from long read sequencing data. What do we do there? We take the long reads. These are these gray bars. And first of all, we mask anything that is not a cytosine, because we do not want to phase the reads based on SMPs. We want to phase the reads based on DNA methylation. So we mask anything that is not a cytosine, and then we remove non-variable positions. So we will basically select along the genome chunks of the genome that are very CpG rich, so they have a lot of CpG density, and where there is a lot of variation in the CpGs along the reads, because without variation, there is no clustering that we can do. And then here we basically hijacked an existing algorithm that what it does is phasing of polyploid genomes. This algorithm, it's called N-phase, and it phases, I don't know, for example, potato. It's a tetraploid, and you need to do phasing with a special algorithm because it's a tetraploid. It's not a typical diploid. So we hijacked this algorithm, but now our variation are not the SMPs, but our variation are the cytosines and their methylation level. So what this algorithm does, it takes any two reads that are overlapping anywhere in the genome, and it scores how similar they are based on the cytosines on those reads. If two reads are very similar, they are going to be condensed into a consensus sequence. And then this consensus sequence, it's put back into the pool of reads. And you keep on comparing pairs of reads and pairs of consensus sequences until you hit the point where there is nothing that can be merged anymore, because all the reads are too dissimilar among them. So then the output of this algorithm is the groups of reads in every one of these areas in the genome which are rich in CpGs. So the groups of reads in that region and their consensus methylation profile in every one of these clusters. So we first tried this method by using publicly available data. This is 3 different breast cancer cell lines for which we know their origin, but we mix them in a single data set. And then we ask our algorithm, can you deconvolve those reads? And can you tell me what clones are present in the data? So here I'm showing you an example of chromosome 6. It's around 4,000 base pairs long, and every gray bar is one of these single reads. And I have, because I know where the reads come from, the program doesn't know it, I've grouped them already by their cell type of origin. And the red dots are methylated cytosines, and the blue dots are unmethylated cytosines. So we applied our algorithm, and this is what we got out of it. We got 3 clones out of it, and the 3 clones kind of correspond perfectly with the 3 cell lines that we put in. So we kind of convinced ourselves, we tested, of course, many more regions than just one region in the genome. We convinced ourselves that we can actually disentangle epigenetic information in a set of long nanopore reads. So then we went into a real sample with a collaboration at the LMU Clinicum, at the clinic hospital, which is right next to our building, actually. It's super convenient. And we collaborated with Oliver Weigert, who's a clinician scientist. So he sees his patients, but he also takes samples from his patients and does a lot of experiments on that. And he had Minayon sample from a mantle cell lymphoma that he had in the clinic. So we took that sample and we analyzed it with our method, and we, of course, discovered a lot of variation. And the first question was like, okay, how can we internally validate somehow that there is some truth on what we find? So the first check that we did is that we validated based on imprinted genes. We went onto a website where we could download a catalog of different imprinted genes. We checked, okay, for what of these imprinted genes we have coverage. And we had 70 imprinted genes for which we had enough coverage. And for 51 of them, we found these beautiful 2 methylation clusters along the genome. And this is an example of that. Again, every one of these long lines is a long read. And as you can see here, this green cluster has very low methylation at that imprinted gene, and the purple cluster has very high methylation at that imprinted gene. So we tested that, and we are good. All of the imprinted genes show the typical high-low methylation. And of course, the epiclones also correspond to the epialleles, which is what we would, sorry, the alleles, not the epialleles, which is what we would assume if we are finding imprinted information. So what about now discovering new, previously unidentified heterogeneity in the sample? I told you that initially in the algorithm, we need to select the regions that we are going to look at. And we selected in that specific data set around 1,600 regions that were CpG dense and had a lot of variation. From those regions, 1,200 of them had 2 methylation clusters discovered. This is where these imprinted regions were, for example, falling. Then we had very few number of regions that had more than 2 clusters covering. And there were some regions that, despite the fact that were CpG rich and had a lot of variation, the method said, sorry, I just cannot deconvolve it. There is no obvious pattern of methylation there. So we decided to look at these regions here. And this is actually what we are doing as we speak. These are a few examples of those. We really don't know what they are. Here you have one of these clones where on top it's a lowly methylated clone. At the bottom it's a highly methylated clone. This track here is a promoter and this track here is a gene body. So we see that we have these beautiful 2 clones exactly at the TSS of that gene. We really don't know why. What are these 2 clones? Another example here, this is inside of a gene body. Again, a methylation clone here, which is highly methylated, a methylation clone here, which is lowly methylated. So what we are now asking ourselves is, where is this heterogeneity coming from? Is it cell type that we are looking at? Is it maybe allelic information that we are looking at? We are talking with people that are specialists in mantle cell lymphoma. They told us there is also a lot of drift at the level of DNA methylation in these diseases. Maybe this is what we are discovering actually. So we are trying to correlate it with different epigenetic marks and what could this heterogeneity be? So this is what we are working on at the moment. So for that part, what I've told you is that even without having the single cell DNA methylation, you can still use this long read data to actually discover heterogeneity inside of your sample. We've tested it on a kind of a boring data set, and we show that indeed we can deconvolve cell types. And then we are testing it now on a mantle cell lymphoma to try to uncover unknown heterogeneity in these samples. And this is work done by Johanna in my group. Good, so now I go back to this picture of the cell and all of the different layers. And the next question that came to us in the group is like, okay, what else can we obtain from that data type? So we can get to the cell types, we can get to the regulation, but is there something else that we can still leverage from that data modalities? And what it occurred to us is that when we measure the ATAC-seq or the bisulfite sequencing, we are measuring over and over the DNA of the single cells. So obviously, if there is any information about copy number variations, we could also be able to obtain it with the single cell epigenomic data. And this is what we did. So we use epigenomic single cell data to call copy number variations in populations of single cells. Classically speaking, if you're familiar with copy number variation calling, you can obtain these beautiful, amazing pictures here. This would be this typical classical measurement techniques, karyotyping or interface fish. It's very nice, but they have very low genetic resolution, and you do like 1 cell at a time. So obviously it's not very much high throughput. Bulk techniques are also very useful. There you really get a lot of genomic resolution along the genome, but obviously you do it for bulk methods. So you cannot really get to the individual single cells. The gold standard would be the single cell DNA sequencing, where you measure the DNA of every individual single cells. And there you have the genomic resolution, and also you have the single cell resolution. So we did that already quite a few years ago. We developed a method that we called aneupfinder for finding aneuploidies in individual single cells with single cell DNA sequencing data, because obviously when you map the reads on the reference genome, if you have a gain, you will see suddenly 1.5 more reads mapping in that position. And if you have a loss, you would see half the number of reads. So obviously this data can be inferred like that. Now, again, single cell DNA-seq is up here. And here you have these much more functionally relevant measurements that nowadays we do much more often, single cell RNA-seq and single cell ATAC-seq in particular. So we decided to try with single cell ATAC-seq, how good can we leverage it for calling copy number variations at the single cell level. So what did we do? We devised an algorithm that we called epi-aneupfinder, compared to the ANO finder that we had developed before. The step number one is that we count the reads in bins. We have 100,000 base pair bins in the genome, and we count how many reads are falling in every one of these bins. And we get a plot like that, where in this axis, sorry, it's super small. This axis here is how many reads are in one bin, and every dot here is a bin. And here you have all the chromosomes separated by these little white spaces here. That's the first step. We obviously need to remove a lot of like bad regions in the genome, telomeres, so on. Then we apply a binary segmentation algorithm using the Anderson-Darling distance that will identify breakpoints in the genome, positions where we have changes in distribution. So imagine this little toy example here. Every dot is a bin. This would be the genome in this direction. What we do is that we partition the genome in many different positions, basically like per bin, and we ask at every position how different is the distribution between left and right. And the moment where we hit like the sweet spot where there is a copy number change, that's where the distribution, the distance is going to be maximized. And we use the Anderson-Darling distance because it emphasizes the extremes of the distributions. And in ataxic, there is a lot of dropout. So it actually helps us a lot to use the Anderson-Darling distance. Good, so with that, we identify breakpoints. And then the question is, okay, given those segments, what are the copy numbers in those positions? For that, we take the average of the genome, and then we do like a statistical test, how likely it is that this region is gained or lost, or it's just normal. And then we color it in green for normal, in red for gain, and in purple for loss. We've tested this algorithm very extensively, both with real samples from collaborators and with more boring kind of cell line samples for which we know the ground truth. And I'm going to show you more like the boring examples here. Sorry about that, just to convince you that it actually works. So this example here is a cell line. It's a gastric cancer cell line, which is a very interesting one because we know that there is a lot of clonality, heterogeneity inside of the sample. So we are supposed to discover a lot of different clones there. We have more than 3,000 cells, and this is what we get when we apply epi-ANO finder. So here, every one of these rows, these tiny little rows, this is the individual single cells. Here you have the chromosomes as columns, and the green parts of the genome are the normal ones. The red ones are the gains, and the purple are the loss. So we see that, yeah, sure, we find gains, we find losses, we find normal, but is that true? So for that, we took another publicly available single-cell DNA sequencing data set for which we analyze, we call the copy number variations, and then we compare the ataxic copy number variations to the DNA copy number variations. And I hope that I can convince you that ataxic is obviously much more noisy. The data is much more sparse compared to DNA sequencing, but we see a very similar profile. So we see, for example, in chromosome 3, long gains versus short gains that are also seen in the DNA-seq. We see these 3 bands of losses in chromosome 4, and so on and so forth. And these we can obviously also quantify given different metrics. We've tested it in many other samples, for example, the HCT116 cell line, for which we also had a single-cell DNA-seq data set. And very importantly, we also tested it for normal samples, euploid samples, where you're not supposed to observe any copy number variations. This is for brain, human brain, and we are happy because we can say, yes, indeed, if you have a sample without copy number variations, we find a little noise. But definitely you would not look at that and say, oh, I see a copy number variation in whatever chromosome. So we think it's also important to be able to use it as a kind of a negative control. So this is this layer here where we can use this epigenetic information. It also works with bisulfite sequencing. I won't talk about that, but we can use these layers of information to call copy numbers. But actually, there are a lot of methods that do copy number variation calling with single-cell RNA-seq. And now we have a lot of single-cell multiome data. So now for the same single cell, we have the attack and we have the RNA. So why not try to leverage both levels of information to get to even better copy number variation calling? So what we first did for that is that we first compared how well do the copy number variation calling from single-cell RNA-seq work compared to the copy number variation from single-cell ataxic. So we choose for that 3 different copy number variation callers from single-cell RNA-seq data in first CMV, Casper, and Copycat. There are many others. We chose these 3, and we tested it again on the SNU 601 cell line, the one that I've shown you before. What is important here for the copy number variation calling from single-cell RNA-seq is that you always need a ground truth. So RNA-seq is obviously very much confounded with the expression of the individual genes. So what you always need when you do an analysis with copy number variations is that you need to be able to normalize by a sample that has the same cell types, but has in principle no copy number variations, which is on itself always very tricky, because how do you know that that sample does not have copy number variations? But it's a prerequisite in all of the methods that exist. So this would be a profile of a single cell RNA-seq row count matrix with all of the different chromosomes. And these dots here are again like per gene, the reads mapping to them. And then you normalize it by a similar cell type for every bean. And you obtain a profile like that, where then in principle, you should be able to much better identify positions where there are real gains and it's not just an expression increase at that position. So for that, we needed to then find for the single cell RNA-seq a data set that contained the test, SNU 601, and also a reference, a bunch of reference cells from a normal gastric sample. So here is now a screenshot of a small part of the genome. The top part here, this is epigenomics finder, SNU 601 cell line. This part here is whole genome sequencing, also for SNU 601 cell line. And here I'm showing you like the suitable profiles for every one of these methods. In red is the whole genome sequencing. In kind of this booger green is the ataxic. And then you have InferCNV, Casper, and Copycat. This is just a screenshot, but to give you a feeling that every method kind of calls something slightly different. Yeah, so some methods identify huge losses here, for example. Again, here, it's a little very heterogeneous, let's put it like that. We can also quantify it genome-wide. We use many different metrics, but I'm showing you here the correlation between these profiles. And what you see, we compared every one of these profiles to the whole genome sequencing profile that we assume is the ground truth. And what you see is that this value here, which is epigenomics finder, is very high. And the values from the copy number variations from single cell RNA-seq data, they are much lower. Yeah, so they contain much less information. However, so this would be this conclusion here. However, they're not completely uncorrelated. So it should still be possible, if you have a multi-omic data set, to leverage the two layers of information to get to a better result in principle, because there is a certain correlation between the RNA and the copy number variation profile. So in view of this multi-omic data, if we want to combine the RNA and the attack information, what would be the best RNA method that we should use to be able to combine the two in the best way? So this prompted us to the next set of results, which is, okay, let's benchmark all of these copy number variation colors from single cell RNA-seq data. Let's discover what are the features that really makes it a good method that then we can include to our multi-omic data. So there we have much more methods. We have 6 of them, and some of them have different flavors, you know, whether they only use the expression or also the allelic information and whatnot. And this is more or less what they are based on. This is whether they only use expression or if they also use the genotypes, meaning the allelic information, how much of every one of the different haplotypes I have present in a given position. The output, some have very, very high resolution per gene. Some it's more like per chromosome arm. Some give it to you per single cell. Some give it to you per subclusters of cells that seem to be more similar to each other. And the majority of them use an external reference. So you always need to provide a set of cells that you know are diploid for the same cell type. Some of them here that say yes, they give you the option that the method will identify the normal cells inside of your tumor sample and will use that internally for normalization. But the normalization always needs to be there. We tested it on a lot of different data sets. We used 14 different data sets here. A lot of them are cell lines, because for those we have a ground truth. So these are easier to deal with. But we also wanted to include a primary sample that was a little bit more challenging, because there you need a ground truth for the same primary sample. So we basically looked online for data sets where we had the single cell RNA-seq and also some kind of whole genome sequencing or whole exome sequencing for the same tumor, which was much more complicated. And that's why we only have 2 of these samples here. And then we also tested diploid cells, because we want to know, can these methods also tell me, no, this sample is fine. There are no copy number variations present there. So we think this is also important. All of these data sets were produced with the 10x technology. Unfortunately, we would like to test what happens with SMART-Seq2, for example. One of the data sets was 10x multiome. And in all of them, exactly, we had a ground truth to know what is the real copy number variations that we should be expecting there. So we benchmarked these methods based on different types of outputs. And one of them, which is probably the most important one, is the comparison to the ground truth of the whole genome sequencing. So here I'm showing to you, on the first row is the results from the single-cell whole genome sequencing profile. So that would look something like that. I aggregate all of the cells together because all the samples come from different origins. So it's not like for the same single cell, I have the RNA and the attack. Yeah, so I do a pseudo-bulk aggregate of the ground truth. And this is this line here. So these are the different chromosomes here at the bottom. And here in blue, it's losses, and in red, it is gains. Now, for every one of the methods that we tested, we get very different outcomes. For example, for inverse CMV, we get really per individual single cell a level of how gain or how lost it is. So in that case, we needed to then impose some cutoffs and say, well, from then on, I call it a gain, from then on, I call it a loss, or I aggregate, or yeah. So in the case of inverse CMV, it was like that. In the case of numbat, just to give you another example, there you get different subclones for the cells, and it tells you for this subclone here, it had 10 cells and it has this profile. And for this subclone here, it had this other number of cells, it has this profile. So because it was difficult to deal with that, we decided to pseudo-bulk everything and to just compare the pseudo-bulk results. And this is what you see here for the SNU 601 cell line. So the whole genome sequencing on top and all of the different methods at the bottom. You see there is quite a lot of variation, and we can, of course, quantify that. We quantified it using different metrics, and this is what I'm showing you now here. This is the F1 score, and all of these dots here are the 14 different data sets that I told you that we tested. And these are the different methods here. So numbat, copycat, and so on. We try to order them from the best to the worst. But what you can obviously see is that there is a huge amount of variation between data sets. So even the best method can perform extremely well for some data sets, but there are some data sets like down here, this red dot, for example, it's really a bad performer for no matter what method you're looking at. We did F1 scores, we did many other type of metric quantification, correlations, AUCs, whatnot. And we always see this huge spread between the different data sets. And what we also see is that there are some methods that are very good at calling gains, for example, but they are rather bad at calling losses. So there is like a trade-off, apparently, between what the method puts the most energy on. We try to then correlate what are the data set characteristics that are the most important to determine is it a good performance data set or not. And what we saw was we saw a clear negative correlation with the number of dropouts that you have in your data set. So the coverage seems to be very important, but also the amount of the genome that is a gain or a loss. So if you have a very small part of the genome which is gain or lost, then you're doing badly here. So this was the comparison with the ground truth. We also tested other things. For example, how good are these methods at identifying euploid cells, samples with nothing. First caveat here, we need to normalize by something. So we decided to take T cells, and we need to normalize by T cells. So what we first did, we took a data set, we split it into two. T cells, we wanted to test if they had aneuploidies, T cells that we assumed didn't have aneuploidies. And this was the result that we obtained. We were very happy. All of the methods kind of said, yeah, no, it's fine. This sample is euploid. But this is obviously not very realistic because we took the T cells and we split them into two. So then we thought, okay, let's do something that is more realistic. Often when you have a sample with a tumor, you will take the immune component and you will assume the immune component is fine, and I will normalize my cells by the immune component. So we took the T cells and we normalized them by the monocytes. And we got quite a lot of copy number variations. Then we did something that also a lot of people do, which is to say, well, my sample is all messed up, but I have a healthy guy there with the same cell types, and then I could use it for normalization. So we took CD4 T cells from another sample, and we used it for normalization. And we also obtained a lot of copy number variations. We can quantify that with just like a root mean square deviation. And what we see is that half CD4 T cells was the best result. But as soon as we deviate from that, nearly all methods identify a lot of copy number variations, except for numbat, that takes into account the allelic information also. And this seems to really be very powerful at the level of identifying euploid samples. Good, so what I've shown you here is that actually you can use single-cell ataxic data for calling copy number variations, and you do it even better than if you use single-cell RNA-seq data for the individual single cells. What we think is really cool is that now you're really able to add an extra layer of information to any single-cell ataxic data set that you already have without having to spend an extra cent. So this is very useful, we think. And this is an R package that we released last year. And then what I also show you is that, the single-cell RNA-seq copy number variations, it's very much dependent on data set performance. So it's a little bit difficult to decide what is a good method and what not. There is a real trade-off between identification of gains and losses. And there are still, however, some methods that seem to be the best ones, particularly NumBut, that performs both good at the comparing with the ground truth and also at identifying euploid samples. And the pipeline is also on GitHub. And there is a poster, the poster of Katarina, a postdoc in my group that has led the majority of this work, that is also in the room. If you want extra information, she's also there for you. And that's all I wanted to tell you today. I want to thank my group for all the work and all of the collaborators and the affiliations. Thank you very much. I can't repeat the question. Yeah, I can do that.
Fantastic. I mean, this is
Thanks.
Really powerful what you can do with this. So in terms of the single cell ataxia and then copy number variation detection, there were a couple of chromosomes where systematically it seemed like we are unable to detect things that the whole genome sequencing is detecting. And then it may be relevant to the accessibility profiles of those chromosomes compared to the local background. So are you experimenting with using more like local chromosome-specific or more like region-specific backgrounds to normalize against to be able to?
Yeah, so the question is that when we compare to whole genome sequencing from the single-cell ataxic for the copy number variations, there were some regions that we seem to lose, and if we can concentrate on these regions by using a different normalization, for example. For the ataxic, we did not normalize. This was really just number of reads per window, so we do not normalize. What we did on those regions is that we were checking if there were not maybe some coverage issues. Mm-hmm. And what we also tested, but it was not very successful, is whether we could identify locations in the genome that may have some gene clusters that may be highly open or highly expressed in a given cell type, and that then would always be seen as a spurious peak in that given location there. This is something that we are still exploring, also for the single-cell RNA-seq part. Yeah.
Very good talk. I'm just wondering, is there any ability to detect micronuclei or other kinds of stranger versions of copy number variations?
So the question is whether we can also detect micronuclei or other types of copy number variations, or parts of the genome that are present or not present. We never tested it, so I cannot give you a definitive answer. But my feeling is that you could detect copy number variations on anything that you put on your reference genome. So if you map on that reference genome, then you should be able to detect whether you have copy number variations or not. What I kind of went over it, but I didn't put much emphasis on, is that we detect normal gains or losses. We do not really detect trisomic, tetrasomic, pentasomic, because the ataxic there is so sparse that you can't just say there is more or there is less, but not there is 2 times more, 3 times more, 4 times more. So I guess like for example, circular DNA that may have a lot of copies, you may be able to detect, yes, it is present, very present, or not very present, but I don't think that you would be able to say I have 100 copies of it or 20 copies of it.
I'll take the last question. I was wondering, because where you were looking for the ground truths, if you look into strand-seq data by Ashley Sanders, because that seems a good start.
Yeah, so actually, I know Ashley from when I was in the Netherlands. So some of the DNA sequencing part, we didn't do it together, but we were talking quite a lot together. But strand-seq is very sparse, and there are not a lot of data sets available for strand-seq data. So we didn't really check how much it correlates. I mean, I guess what would be cool is that, so with strand-seq, you would be able to say, oh, I have like a sister chromatid exchange at that position. We could see whether we could see it also as a level of a copy number, but we didn't really explore it. We work a lot with publicly available data. So for comparing modalities, you're always dependent on having the same cell type that has been profiled with a tag and with RNA and with transcript and with DNA. And so it gets smaller and smaller, the amount of things that you can test. Yeah. Thank you very much.